Well, thank you everyone for joining us for the session on ataxia biomarkers and why they are important. I am Dr. Hayley Mcloughlin. I am going to be co-presenting this session today with Dr. Puneet Opal. And I just want to quickly show this slide about the disclaimer on some of our presentation information. And also our disclosures. So Dr. Opal from Northwestern University has no conflicts with material presented. He does have research support from Biohaven and royalties from UpToDate. I, from the University of Michigan, have no conflicts again with the material presented in this talk. We have research support from Ionis, Biogen, and Unicure, and I consult for Lacerta. So without further ado, we wanted to get into it. So we are approaching a pivotal moment in SCA research, where drug development and preclinical models have shown potent effects. And there's just momentum forward for SCA types towards clinical trials. As you may have heard in the clinical trial readiness session prior to our talk, there are many components that go into clinical trial readiness. And so what today we're going to talk about is some of the challenges that need to be addressed for the clinical trial readiness. As we are approaching, as this slide indicates, many different modalities that target SCA known dominant and inherited disorders as displayed in the table. In particular, two of these are actually in phase 1 clinical trials. We have SCA2, which is approached with ASOs against ALS mutations, as well as SCA2 patients. And just in the last month, there has been Biogen on phase 3 clinical trials for ASOs in SCA3. So among those challenges for clinical trial readiness, there are issues with low SCA3 disease prevalence. We know that global prevalence of this SCAs is estimated between 1 in 6 and 100,000 people. This low prevalence of SCA3 SCAs will make it a challenge for recruiting sufficient patients for any given trial. Now, also there's clinical heterogeneity, and this just basically means that across all of the SCAs, there are many different clinical features that differ, even though there is a shared common mutation for many of the dominantly inherited ataxias. In consistency with that, we have genetic heterogeneity. So within each disease type, there is also differences that occur across individuals, which is indicated by both the disease penetrance, the age of symptom onset, the rate of the symptom progression, and all of these may really complicate our ability to monitor progression and the response of disease therapies. Finally, we have a lack of objective disease measures that is something that is a real big challenge in the field for clinical trial readiness. So industry and academics, nonprofits are all really trying to address these clinical trial readiness by generating well-validated preclinical data. This is in cell models. This is in animal models. We are also looking forward to updating and addressing our clinical disease measures using wearable sensors and also our clinical rating scales. We are using discovery platforms and developmental strategies from biofluids, as well as imaging biomarkers for disease, and we're going to talk about those further. And then also there's a lot of efforts in just strategic trial design, which you may have heard about earlier, with patient recruitment efforts, statistical modeling, and then finally, of course, funding support for all of these different engagements in this clinical trial readiness process. So we can't cover everything in a 35-minute talk, and we want to get on to the Q&A, but what our goal is for today is to talk about how biomarkers can really assist in this clinical trial readiness. So some of the topics we're going to cover in this talk are, what is a biomarker? Why are biomarkers valuable? What are the different types of biomarkers and how are they being used in SCA research as well as in the clinic? How is data and samples collected? And then finally, we'll get to how you can help. So let's start with the basics. What is a biomarker? The definition according to BEST, or the Biomarker Endpoints and Other Tools resource, which is developed and defined by the NIH Biomarker Working Group, is that a biomarker is a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, and responses to an exposure or an intervention. Some of these characteristics can be defined through functional assessments, molecular assessments, and imaging assessments, of which we're going to go into further detail in this talk. But first, we need to address why biomarkers are valuable. So biomarkers are valuable, or said another way, how can they be utilized? A disease biomarker is a disease state biomarker is something that indicates when a treatment will be most effective for a disease. A target engagement or a pharmacodynamic biomarker, these can indicate whether a treatment is actually targeting the disease gene of interest or the target of interest. So for the target engagement biomarkers, and the pharmacodynamic biomarkers is whether it's eliciting the proper response or quite likely the incorrect response, leading to any sort of potential side effects or unwanted issues that we want to avoid. So there's going to be 3 separate types of biomarker assessments that we're going to be discussing today. And the first of those we're going to get into is the functional assessments. So these are clinical assessments or wearable sensors for daily tracking of movement. There is a full battery of functional assessments that has been utilized in SCA research. Among the most common is SARA, or the Scale for Assessment and Rating of Ataxia. So SARA is a measurement of motor coordination through which there's 8 separate motor coordination tests that are assessed by a clinician. They're going to be gait, stance, sitting, speech disturbances, finger chase, your nose to finger test, a fast alternating hand movement, or a shin and heel slide, all of which have a rating score from 0 up to 4, all the way up to 8 for gait in that case. The higher the score, the more impaired you are in the task. And again, if you add up all of the scores across these 8 different motor assessments, you'll get a total ataxia rating score, meaning 0 is no ataxia in a patient, whereas 40 is the most severe ataxia that can be seen. Now, the utility of SARA in clinical trials was recently assessed when combining 6 different studies across almost a decade, over a decade of work. There's over 1,000 SCA patients that were enrolled in these studies, and they pulled together the SARA scores over a 1-year annual assessment. So if you plot all of those samples and normalize to 0, what is the average SARA change over an annual visit? And we know that SCA1 across the dominantly inherited ataxias is the most progressive, whereas SCA2 is less progressive, only occurring about 1.4 SARA score change over 1-year time point, and SCA6, which is least progressive of the dominantly inherited ataxias, again, less than a SARA score. But how can those actually be utilized? So to translate this information, we can use this to inform trial design, as what you might have already heard. And this can inform how many patients we actually need to enroll in a study. I think this really elicits why biomarkers are necessary, because as of right now, to understand in a 1-year study, using just SARA alone and showing a 50% reduction in the progression, so in this case, it would be a 0.9 on a SARA score reduction, you need to enroll 92 patients. 92 SCA1 patients is a challenge. We have 1 to 6 patients in 100,000 of SCA patients in the world. And so the low prevalence state makes it really a challenge for any sort of recruitment in a trial, and really informative of how these are working, or how these are moving forward in clinical trial readiness. So what we're looking for is other opportunities as surrogate biomarkers that can also collaborate or inform with SARA, or even as pre-manifest state before any symptom onset. So one such is the wearable sensor technology, which is being discussed in both natural history studies as well as possible enrollment in future clinical trials. If you go to clinicaltrials.gov, there's a study that is currently recruiting SCA1, 2, 3, 6 and Friedreich ataxia patients and age-matched healthy controls. In this IDEA study, or instrumental data exchange for ataxia study, the research study is testing whether body-worn sensors are able to measure movements in simple tests like coordination. And so in order to evaluate the progress and the severity of ataxia. And so one paper that was recently published this last December in 2021 was an outcome of this data set, in which they assessed 163 SCA patients across different dominantly inherited ataxias relative to healthy controls and found that this toe angle, so if you walk in a straight line and you have a sensor around the ankle, you can see that the degree of toe angle out actually can inform whether you have the disease or not, and can separate that in both us and can correlate that with the severity of your score in reference to SARA. Another test that is also informative is this double support. So when you're walking, how often do you have to have both feet on stable ground versus lifting off? And so those two different modalities from a wearable sensor might be very informative in collaboration with SARA to help us power a future clinical trial study. Importantly, this wearable sensor technology is also more sensitive than SARA. And so in this one study, they did a prodromal state, so pre-manifest prior to ataxia symptom onset, as noted by SARA. They can distinguish pre-manifest patients from manifest ataxic patients. This is early days. This is one study in movement disorders that came out last December. Future work, as with most scientific articles, have to be validated in an independent cohort to really generalize these results moving forward. But it does show the utility of wearable sensors in the future. Now, these functional assessments like SARA are really limited to only patients with ataxia symptoms. But what about the biomarkers that distinguish SCA patients prior to ataxia onset and then also correlate as disease progresses? So for that, we're going to look forward into imaging assessment opportunities. And so there are other biomarkers such as using an MR machine in which you can detect the brain size as well as different metabolites in a non-invasive way. So you can repeat measure patients in this imaging modality. And some of the different output data that we can get from this is MRI. So looking at both the different types of tissues in a brain, how the volume is changed over time. You can look at the metabolites that are changed in a brain, as well as tagging things specifically in a brain to assess whether they're changed, like a dopamine marker. And so one example of this is MRI, magnetic resonance imaging. So MRI is using a scanner with a really strong magnetic field that generates pictures of your brain or of body organs if you're using it for other modalities. And so in this case that I'm showing on the right, what researchers had done has engaged in enrolled SCA 1, 2, 3, and 7 patients, as well as control samples patients, and looked at their brain volume in both the cerebellum as well as the pontine brainstem region. These are two affected brain regions in many of these diseases. And as you can see by the differences in change over a 24-month period, we can actually quantify in a non-invasive way how in a neurodegenerative space these volumes actually get smaller over time. This is one of many different studies out in the literature that support this, but overall there's mounting evidence that indicates that conventional MRI, such as what I displayed here, is really able to consistently show cellular changes both at clinical onset and actually prior to onset of ataxia symptoms. Another utility of scanners is you can use magnetic resonance spectroscopy. So this is a way in which you can actually isolate specific areas of a brain, and you can look at spectral scans within that brain region for metabolite changes. And you can reference those to control population across different brain regions. And so in this right-hand picture here, I'm showing again multiple SCA patients, as well as reference control from 3 separate brain regions. Again, 2 in the cerebellum, very vulnerable brain regions in these diseases, as well as the pons, like what we saw in the MRI. And across multiple studies, again, the MRS field has used this to inform both levels of neural markers like total NAA or glial markers, the cells that support the neurons in the brain, total choline markers, and can show that they are significantly correlated with ataxia rating scores like SARA, as well as the activity of daily living scores in subjects with SCA. We can take this one step backwards and actually go back to any sort of preclinical development of any sort of therapeutics. And this is an example from my laboratory We were using, again, an MRS machine, to image the spectral changes in a mouse. And so again, we can isolate cerebellar regions, and we can show that total NAA, so the changes in the neuronal metabolites, is actually with a wild type here decreased in disease. But when you treat it with an ASO, much like what we will be enrolling in patients in the future, you can rescue this metabolite change, so neuronal rescue. I think that shows the utility of both the assessments for the clinic as well as imaging assessments. And now I'm going to hand it off to my co-presenter, Dr. Puneet Opal, to discuss the latter half of this presentation.
Thank you very much, Dr. Mcloughlin. So you've heard about the functional assessments where you're looking at the patient, and then Dr. Mcloughlin talked about the imaging assessments, which are non-invasive ways, and now you're assessing the brain. I'm going to talk about fluid assessment. So we're not looking at the brain, not looking at the patient, but actually we can get some pretty good clues about what's going on. At least that's the hope with the spinocerebellar ataxia as well. So when we talk about fluids, what can we get from fluids? We can get DNA, which is basically the information of a cell, and Dr. Mcloughlin mentioned that earlier. There's RNA, which comes from DNA, and then finally the RNA makes the protein. Now, all three of them can be quite valuable, and we can get fluids from a variety of different sources. We can actually get from blood. You know, as you go to the doctor, you just take a prick, and they can also fill a tube of blood for biomarkers. You can also get a fluid from the spinal fluid. It's a little bit more invasive, but that really gets to a fluid which is close to the outer surface of the brain. And since it's sort of touching the brain, we might get better clues to what's going on in the brain. It's a little bit more invasive. And then probably the least invasive of all, and believe it or not, we can actually get some clues from urine samples also. And what are all these clues that we can get? From DNA, we can actually get the genetic diagnosis, but most of you know that the genetic diagnosis is basic. You already know your genetic diagnosis. With biomarkers, what we want to do is we want to see if there are any other things in your genome which could be affecting the disease. So we know that some people progress faster or slower, but we don't know why. It could be something in the environment, but it could be also because of some other genetic factors. So these are called SNPs, basically single nucleotide polymorphism, which vary between people, which could be associated with some other DNA regions which are aligned with disease progression and onset. With RNA, you can actually get a feeling of what's going on in cells, and if they are not behaving well, they might have a different expression pattern, and there are different types of RNA. There's a small RNA and a messenger RNA, but those are relatively details. And with both DNA and RNA, the good thing is that they are very good assays to amplify the signal and also sequence the signal. So we have a very accurate way of looking at what's going on if we can get samples and address DNA and RNA. The third on the list is protein. Protein actually is the final outcome, and if there's a problem with the cell, you're going to have abnormal proteins, which either might be leaking out into the bodily fluids. And for that, we have proteomic analysis, or candidate proteins can be discovered by using tools like antibodies, a test called ELISAs or single molecule analysis. And this tells you actually that analyzing protein is actually much more difficult because you need to, there's no really way to amplify the signal except by basically amplifying your detection technique. And these can be more complicated. But if you can get an analyte and really understand what's going on, it might be a very good biomarker as well. So here's an example of what one would do with blood, for example. With blood, you can actually get plasma by spinning it down, which is like the liquid phase. Red blood cells are the form a majority of the cells, but red blood cells actually don't have nuclei, and they don't have DNA and not much RNA either. So what one really goes for when you want cells is actually you go for the white blood cells, which are in the buffy coat, and they're sort of in the junction between the spinning down of the red blood cells at the very bottom and the plasma at the top. Now, similar things can be done with spinal fluid, and I told you spinal fluid surrounds the brain and the spinal cord. And for those who have not had a spinal tap, I just want to point out that it is more invasive than a blood tap, removing blood from your body, but it's also not that invasive too. It's basically a bedside procedure. What one does is the physician or the person doing this will have the patient lie down in a position here. It's called the left lateral decubitus position. And the area is numbed after being very clean and sterile. And the area is numbed basically the way the dentist would numb your mouth before taking away a tooth, for example. But here what we're doing is we're numbing the skin, approaching the spinal fluid at a level where the spinal cord actually ends. So there's no possible damage to the spinal cord itself. There's some nerves and nerve roots which emanate, but this is a place where there's just these nerve roots and there's a lot of space by which we can put an injection needle, or a needle like that, actually, it's called a spinal tap needle, and withdraw a sample fluid for biomarker assessment. So I just wanted you to sort of see what it looks like. There are online videos and YouTube videos, which you can go into more detail, or we can address in the question and answer session later. So we mentioned the goal of biomarkers, but the real goal of biomarkers with these fluid biomarkers, I want to emphasize, is really clinical trial readiness. And here we come again to this idea of target engagement. So we mentioned the ASOs as a way to knock down proteins. So it would be nice to see if you can detect that protein, for example, being knocked down in a clinical trial. Then it could be a surrogate marker for disease onset and progression. So this could either be the protein itself, which you're trying to knock down, if it correlates with the disease, or other proteins or other RNA, which might be sort of going along with the disease severity and progression. In both of these, in our clinical trials, we don't really need DNA to be analyzed, because we're really just trying to see what goes on once the antisense oligonucleotide is sort of hitting the targets such as ataxin-1 or ataxin-2 or ataxin-3, the different SCAs, SCA1, SCA2, and SCA3, for example. The biomarkers are also very important because we might be able to stratify patients when we come down to clinical trials. It's always good to compare patients with the sort of same level of disease burden at the outset and compare them once they are either getting a placebo or getting a treatment. And you don't want to have patients who are out of whack, if you will, with regard to the disease onset progression. And potentially these biomarkers, especially the surrogate biomarkers, could help with the inclusion criteria and stratification as well. And I finally want to mention that these are still early days, and whatever clues we get from biomarkers through our discovery might actually lead to unanticipated novel ways of thinking about the disease, novel pathways or modifiers which might inspire new treatments and new treatment strategies. So how would this look like? So this is an example of what a target biomarker would look like. Of course, none of this is in clinical trials yet, but one of our collaborators, Dr. Petruccelli, has identified a way to identify ataxin-3, the protein ataxin-3 in plasma and CSF. And this actually is not so trivial. It actually requires a very high detection capability. And this has the basics have been worked out. The question is whether we can improve it and make it ready for a clinical trial. And what you can see here is that you can see that patients with SCA3 have a higher burden of ataxin-3, presumably because the protein is not cleared as a wild-type protein is. And surprisingly, even pre-symptomatic patients, so these are patients who have the mutation but do not yet show clinical features of the disease by SERA score or other functional rating scales, even they have an elevation of ataxin-3, suggesting that a biomarker can also pick up a pre-symptomatic patient as well. Now, that was an example of a target biomarker. This is an example of a surrogate biomarker. It turns out that there's a protein called neurofilament, or a neurofilament light chain to be more specific. It's a member of a cytoskeletal family of proteins, and they're extremely abundant in the brain, and they're also specific to the brain. And believe it or not, this protein actually can be detected in the blood, which makes it easy without doing even a spinal tap, although spinal taps can show even a more precise level, but you can actually detect this in the blood because the new techniques of detecting proteins have reached a point where we can detect very, very low concentrations when it leaks on the blood. And because it's neuron specific, we know that whatever's happening in the brain is the only way that this will be leaking out into the blood. And here I want to point out, again, from another publication by a group in Europe, where they have found that in a cohort, they basically taken many patients, and they find that the neurofilament light chain is increased in ataxic patients, but also pre-ataxic patients, once again speaking to the fact that you can actually detect this in the pre-symptomatic phase of the disease. This is just showing more of this data in a scatter plot. But the important thing to show here is, in this last picture here, is that it turns out and this is a group of patients with SCA3, that the longer the SCA3 expansion is, so you know that SCA3 is caused by a trinucleotide expansion of the CAG repeats. So the longer the repeat length, we already knew would cause an earlier disease onset and a faster progression. And this neurofilament light chain biomarker actually does reflect that, where you find patients with this very high onset CAG repeats of around 73, showing the highest levels on the y-axis of the neurofilament light chain, and also early age, as opposed to patients who have a relatively lower CAG repeat of 61, for example. Now, I told you about disease progression with the neurofilament light chain, but we know that neurofilament light chains can be detected even in the normal people. It's just not at the high amount, but in normal people also there's an upgrade or increase of neurofilament light chain with age. So the question of disease progression has to sort of be separated from the neurofilament light chain progression just by age itself. And this is an area of research which definitely has to be studied as we move neurofilament light chain from the bench to the clinic in clinical trials. So the data I've shown you so far has been done on clinical cohorts, some of which have been also helpful for the functional assay, which Dr. Mcloughlin mentioned, or some of the imaging assays. But these are some of the acronyms you might have heard of, or they may be on the National Ataxia Foundation websites. The top three are from the US studies. There's a CRC-SCA study, which is funded essentially by philanthropy from the National Ataxia Foundation. There's the ReadySCA, which is an NIH-sponsored study, but it focuses on just two ataxia, SCA1 and SCA3. And University of Michigan has a separate study called BioSCA3, which focuses on SCA3. The ReadySCA, for example, focuses just on the early stage SCAs or the pre-symptomatic, but the BioSCA SCA3 focuses on all the SCAs. There are a few other acronyms which our European counterparts who are studying ataxia are doing. EuroSCA essentially aligns with the National Ataxia Foundation. RISCA is looking at ataxia at risk, and ESME is looking at SCA3 in more detail, much like, I guess, the ReadySCA and the University of Michigan sites are doing here. But I put them here to say that, these patients like you are rare, and we really have to get all our cohorts to really understand. Sometimes we use the US cohort as one side and the European cohort as another side to sort of validate what's going on. And we really try to pool our resources, and we try to have the same ways of doing things, the same SARA scales, the same imaging techniques, and also with the biofluid, the sort of same biofluid ways of collecting our samples so that they are uniform across sites and we can make really good comparisons, especially during the research phase. Our hope is that these comparisons will be robust enough down the road as we move to the clinical setting. Now, why do we need so many different studies? Well, in a way, there's an advantage to keeping the ataxia separate. Ataxias progress at different rates. We know that SCA1 progresses slightly faster, SCA2 and 3 are intermediate, and SCA6 is the slowest. And so it's likely that biomarkers will reflect these different trajectories, or we would actually hope that if the disease is progressing rapidly, we would find biomarkers which would reflect the rapid progression. But pooling these might sort of hide these differences. It also might be too ambitious to study all the ataxias with identifying funding mechanisms in place. For example, University of Michigan had this grant to study SCA3, but it would be very difficult to sort of spread those resources to other SCAs till we get some good data on just one ataxia. And it'll also be likely that clinical trials will focus on each ataxia individually. So there's probably going to be one trial for, say, SCA1, another for SCA3. It's just easier to deal with different SCAs rather than pulling them all together. So, with this, I hope that you sort of got an overview of some key questions which Dr. Mcloughlin mentioned in the beginning, which is, what is a biomarker? Why are biomarkers so valuable? What are the different types of biomarkers? And how are the data samples collected? So whether it's functional rating scales, the imaging, or the fluid, the bodily fluids. So now I want to just end by saying how you guys can really help us here and help the field move forward. So this is an endeavor which requires a whole operation, and the National Ataxia Foundation, for example, has been very helpful in setting up the initial CRC SCA sites, and some of the newer studies have sort of tagged on to that, the ReadySCAR site and the Michigan SCA3 study. And some of these sites, I'm showing you the map so you can get a feel. This is not totally up to date. For example, there's another site right now in Dallas, for example. But there's a geographical spread about this, which is really good, so that patients don't have to travel too far. They might still have to travel, but there's probably at every point within the US, there's a place where one potentially could go to. And some of these sites also, not all, because imaging can be quite expensive. So a few of these sites are set up to do imaging in a very standardized way as well. And those sites are sort of mentioned here on this slide with a sort of a little halo in the middle. And there's overlap between some of the National Ataxia Foundation sites and some studies are being done in only some centers, such as ReadySCAR, for example. Some of our American studies are also being collaborated with in Europe as well, and there are a couple of European sites, so I kept them on this slide. But there's also a whole different set of studies which are dedicated in Europe. And I know this, some of you are international and seeing this from by Zoom. So you might, if you're from Europe, for example, you might have a site which is close to you where you can also donate some of these samples. And these studies are being done, are at least being proposed to be done by an Ataxia Global network. And the Ataxia Global is really trying to pool the US and European groups and eventually cover the entire globe. We might even we're actually aiming to have sites in Asia and Africa and developing countries as well. So that everything is done the same way so we can have more cohorts, more validation, and just more progress. So that's really where I wanted to stop, where both Dr. Mcloughlin and I wanted to stop here with, but making an appeal that if you're not a part of a ReadySCAR or CRC SCA study, please join us, because your bodily fluids or your imaging or your functional assessments will be really helpful, because without knowing these baseline assessments, even before we go into invasive clinical trials of actually testing it, we would want to know how a drug, for example, would change the trajectory of the illness. And we need this baseline information to get that baseline trajectory on which the drug or the placebo will now be tested to see if it really can bring down either the disease onset or disease progression. So I thank you very much for your attention. I know some of this might be a little bit technical, and therefore, please feel free. There's, every question we will entertain, and we can start from the basics, because I know many people might have different backgrounds. So, I apologize if some of this has been very technical or a little bit jargony, but this is a good time to ask questions. So thank you so much.
Hi, thank you, Dr. Mcloughlin and Opal, for your fantastic presentations. Hello, my name is Lauren Moore, and I'm the Chief Scientific Officer of the National Ataxia Foundation. And I'm joined here live by Drs. Mcloughlin and Opal. And we will be taking your questions live, and just a few housekeeping items before we get started. If you have a question, please feel free to post them by clicking the Q&A button at the bottom left-hand side of the platform. If your video currently is in full-size mode, you won't see that Q&A question link, so you'll have to first minimize the video before you can take the Q&A. So we'll start off with a question for Dr. Opal. Can you please describe some of the biomarkers that currently are being used in clinical trials, like the Biohaven trial presented earlier today?
Sure, I think that's a great question. So the Biohaven trial is actually a very interesting trial, because the first clinical trial for a pharmacological agent for the Ataxia in the US. And in terms of the rate, in terms of what people are following right now in the trial, a major component has been the rating scale. So there's a rating scale, which I mentioned, which is the SARA scale. And the SARA scale has been modified for the Biohaven trial to reflect disease progression and to see if the drug, experimental drug, shows any beneficial effects. In other words, that there's less progression of the disease. There's not yet any imaging or blood biomarker currently with the Biohaven, but these the newer biomarkers that we mentioned, both imaging and blood-based, will probably be used in future clinical trials, especially with the example with the ASO trials planned by Biogen, for example.
Great, thank you. So once we do have more biomarkers, such as the fluid biomarkers you talked about earlier, how might that change the way clinical trials are designed? Will it change the length of the trial, and how might it change the experience for the patient?
So it's really difficult to recruit a lot of patients to have enough power. Power is like whether you can see a difference or not. And the goal of biomarkers is basically to help as one additional measure. So maybe we can reduce the recruitment efforts and we can have fewer patients to get a power, but this time the power will be with the patient rating scale in addition to biomarkers. So I anticipate that with the help of biomarkers, we might have first smaller patient numbers. And the second is that maybe we could also have a shorter duration of trial, and we don't have to keep trials for so long because we might start seeing difference in the biomarker, which might suggest improvement, which might allow us to curtail the length of the trial and make it more affordable, because clinical trials are extremely expensive.
Great, thank you. And Dr. Mcloughlin, I understand that you actually volunteered to participate in a natural history study yourself as a control. Can you tell me a little bit about what that experience was like and why you actually chose to volunteer to participate?
Absolutely. Yeah. So one of the studies that Dr. Opal spoke about was happening at the University of Michigan, run in collaboration with myself and some other colleagues at the University of Michigan. And we had a patient come in. We always have an age match, sex match control, of which I actually fit the perfect control match. Now, this was at the very onset at the beginning of our U01 study. And we were at a point in which we're starting to solicit this out to the community and provide information about how these biomarkers can help us out as scientists and also forward thinking through clinical trials. Now, I didn't feel ethically right or bound to solicit this to anyone or tell anyone about providing samples without having any sort of understanding. I had 2 children without an epidural. So for me to go and get a lumbar puncture and tell someone else to go get a lumbar puncture, I felt like I had to do it myself. Again, because I had the perfect age match, sex match, SCA3 patient that volunteered graciously for our study, I went out and did this. Now the experience was as I hoped. It was a half an hour of my time sitting in a room. I got swabbed up, as Dr. Opal explained, sterilized, anesthetized the area. And actually that was the most painful for me because needles going into my back to give me an anesthetic was not ideal. Now the lumbar puncture itself, or the cerebral spinal fluid collections, was very trivial, actually. It was more just hanging out and chatting for about 5 minutes for us to collect the sample. And then I laid down for 15, 20 minutes, got right back up and actually walked back to my laboratory for the rest of the day.
Well, I think I speak for many people right now when I say thank you for participate in your trial yourself, and I'm glad that it went well. Here's a question for Dr. Opal, and it's a good follow-up from Dr. Mcloughlin. So are samples such as the spinal fluid, blood, and saliva that are previously taken from a patient such as Hayley, are those saved or in some way available research, or do new samples need to be collected every time?
So currently the samples which, for example, are being collected by CRC-SCA are being sent to a central repository. It's called BioSend, and it's located in Indiana University because that's where the affiliation is. And these samples are currently accessible after peer review because these samples are quite precious because only so much blood or spinal fluid is taken at any given time. And so there's a little peer review which goes on before these samples are distributed, either to industry representatives or other investigators, typically at universities or nonprofits trying to study these. The goal of eventually is that we will have ongoing, so it will be replenished, but you're right that it's a pretty precious sample, so sometimes you might have to repeat or get additional samples. Also, there's longitudinal data. So the sample you might give today, we're going to be looking at a similar sample later on when you might be growing older and the disease might be progressing. So it's good to have longitudinal data because we get different information from that. in terms of disease progression. So when the original sample run out, we might not be able to do the additional longitudinal study because we don't have the first reference group, for example.
Thank you. Dr. Mcloughlin, do you have to have a genetic diagnosis always before you can participate in a natural history or biomarker study?
Sure. So these are actually part of the criteria. So I encourage everyone who's interested in donating or providing samples to any sort of study. All of these natural history studies, at least in the US, are on clinicaltrials.gov that we showed on that last slide. And there is both inclusion criteria, meaning things that you have to be able to provide or work in these studies, to become a participant in these studies, I should say, as well as exclusion criteria. Again, based on your history, these are very obvious inclusion and exclusion criteria. Whether you have a genetic diagnosis is based on each of the different studies. So for instance, ReadySCA, which is in the US, and they were profiling for SCA1 and SCA3 patients, they were also looking for at risk, so pre-manifest patients, which means that you didn't necessarily have to know whether you had ataxia. You just had to have, as an inclusion criteria, SCA1 or SCA3 within your family genetic tree. Now, CRC SCA, which is run by the National Ataxia Foundation, is another opportunity for you to get a genetic diagnosis for blood sampling so that you can define whether you are eligible for their natural history study. Of course, knowing which subject sample you would provide for is always helpful. In a case of the one that we're using at the University of Michigan, which is currently primarily only SCA3, we are enrolling for SCA3 patients, but we're also asking for controls. So whether you are a family member that is looking to also source the control subjects, that is a huge valuable resource for this community because for us to be able to say a normal state and a state that we want to get back to with these blood biomarkers is frankly immeasurable.
Great, thank you. Dr. Opal, are they still actively recruiting patients to participate in these CRC, SCA, and ReadySCA trials?
The CRC SCA is ongoing, and we are always recruiting patients. But again, you have to look at the inclusion and exclusion criteria because they're recruiting patients for some of the common SCAs, not all SCAs. For example, we don't recruit SCA 36, for example. We're typically recruiting some of the more common SCA, which tend to be 1, 2, 3, 7, and so on and so forth. Okay, so you'll have to look at the clinical trials website. In terms of the ReadySCA, we are continuing the longitudinal data, but there's no active recruitment right now for the ReadySCA because that was funded by the NIH, and that the duration of the funding is what has led to some constraints about getting ongoing recruitment.
I'm getting several comments and questions that we're talking a lot about the SCAs here. Are some of the biomarkers that might be discovered through these clinical trials, could those be applied to other ataxias, such as recessive or even acquired forms of ataxia? And I'll let Hayley start and Dr. Opal can finish.
Sure. This isn't an easy answer. Again, rare spinal cerebellar ataxias, or SCAs, are not common enough for us to be able to group enough patients together to say, is this normal states of your biomarkers that we are able to explore? Now, with the natural history studies that we have from the more common SCAs, that accumulation of data that's both at a snapshot in time as well as longitudinal will be a really useful reference for some of the more rare SCAs down the road. In the same sense that the clinical trials that are currently being explored or are enrolling are towards some of these more common ataxias, that information will also provide more natural history or longitudinal information that we can apply in the future to other rare SCAs or rare ataxias that are acquired.
I agree with everything which Dr. Mcloughlin said. I just want to say that there's also ongoing studies which I mentioned about global initiatives. And actually there's a global initiative just for rare ataxia. It's called ARCA Global, which stands for Autosomal Recessive Cerebellar Ataxia Initiative. And so these initiatives currently are trying to get off the ground. And oftentimes what takes an ataxia off the ground is funding. So everybody's applying for different funding mechanisms. So I'm assured that at some point, the rarer ataxia will also be tackled with the same determination and perseverance that we're using for the SCAs. But this is exactly where we want patients to be aware about what clinical trials are going to be there in the future. So that's another reason to go onto clinicaltrials.gov. Can
I also add maybe one more piece to this puzzle? So the one biomarker, biofluid that we spoke about was the NFL, or neurofilament light. Now, we didn't come out with this NFL as a novel biomarker for the SCA field or for the ataxia field. This came from neurodegenerative diseases, and it is a commonality amongst neurodegenerative diseases. And so that kind of longitudinal information that we were provided from other trials, other natural history studies, has allowed for us to then reproduce it in our own community. I think that that is a key point to say that anything that we acquire in one realm can be transferred over. The data is there, it just needs to be explored.
Great, thank you. And this question is for Dr. Opal. So to what extent could biomarkers be used to help potentially even diagnose someone who has been, they haven't been able to get a positive genetic diagnosis for a certain type of ataxia, but they have been diagnosed with some sporadic ataxia or idiopathic ataxia? Could biomarkers actually help with diagnosis in the future?
Well, diagnosis of a spinocerebellar ataxia of the genetic kind, the genetic diagnosis is basically the gold standard. But you raise an interesting point that there are many ataxias which we call sporadic ataxias, and MSA or multisystem atrophy can present in a cerebellar form, and it's called MSAC. That's probably the most common sporadic ataxia, but there are probably others as well. And some of the sporadic ataxias could be also because we haven't yet got the genetic diagnosis. They tend to be repeat disorders because nowadays with whole genome sequencing, most of the genetic ataxia are being found, but the repeat disorders require a little bit more than just even whole genome sequencing, because it actually doesn't get the repeats very easily. So now in terms of biomarkers making the diagnosis, so there are ongoing studies. Some of them are just small university-run studies which are trying to collate blood and collect them for patients, for MSA patients as well. And so that might reflect, for example, I wouldn't be surprised if neurofilament light chain is also going to be seen in these sporadic ataxias, because neurofilament, as Dr. Mcloughlin just mentioned, it was picked up in neurodegeneration. And it's just a marker of the nervous system degenerating and neurons releasing these neurofilaments into the blood. So I won't be surprised if some of the markers will be seen in MSAC or other sporadic ataxias. But frankly, these sporadic ataxias are also diagnosed clinically because they have the neurofilament light chain is going to be seen in all neurodegeneration. So actually the diagnosis of ataxia is made as a clinical diagnosis. It's called a sporadic ataxia. And then we find these, we're following some of the neurofilament biomarkers because they're candidates. In the future, it might turn out that there may be a more specific biomarker for some of these ataxias, and that could potentially, I guess, lead to more of a diagnostic reappraisal of some of the sporadic ataxia and shift to, say, an MSAC versus some other sporadic ataxia. But that we have not yet reached that level of science yet.
Great, thank you. And this is a question for Dr. Mcloughlin. Are there commonalities among the causes of ataxia to make it likely that sporadic patients might improve by some of the drugs that are in development for ataxias?
So that's a hard question to answer, frankly. It's all case-by-case basis. There are gluten ataxias that are caused by gluten ingestion. So there's evidence to support that what we learn from other phenotypes we can use as a phenocopy, or as a how your body is, how your ataxia is portrayed to others, and how that might relate to some of the more genetic ataxias. The other aspects of genetic ataxias is that they're not all equally related. So I spoke towards the clinical and genetic heterogeneity, even within a dominantly inherited ataxia. Now, one of the things that these clinical trials or these natural history studies are doing is they're also collecting DNA samples. And so things that we can do in science is look at what's known as a genetic modifier. So a different change in your genome that either increases your ataxia phenotypes or reduces it. And because of those gene simple changes, it may inform some of the more sporadic onsets, again, that is yet to be discovered. Now, why I think this is important is because, again, like I said, the data is there, we just have to look for it. One patient that we can relate to a simple phenotype that we see, or how that we can relate to with these genetic modifiers, may create this whole new reason to assess all of that DNA samples and ask, are any of these sporadic cases more of a genetic variant? So I would speak towards that, whether the actual drug of choice that we use for the ASOs, say for this Biogen trial that's going to be targeting Ataxin 3, that is less likely, unless there is some sort of linkage or reason to believe Ataxin 3 is also dysregulated in a sporadic case. Those are much more precision medicine in a sense, because they're targeting a specific gene of disease.
Great, thank you. And question for Dr. Opal. So from a patient, I don't have one of the ataxias that's included in the CRC-SCA study. How can I find out if there are natural history studies for my type of ataxia?
So I guess a good place to look up would be the clinicaltrials.gov, because there may be smaller university-based trials which are not yet, I guess, advertised sufficiently that your own physician may know about it. That would be one. The other would be that you can bring it up with your physician, especially if you meet one of your movement disorder specialists. You can ask them to look it up if you are not familiar with some of these database lookups, because some of them, requires a little bit of being savvy with computer and knowing where to look. And another way would be always keep up to date with because there may not be a trial now, but there may be a trial a year from now. So always check. I guess the best place probably would be the National Ataxia Foundation, where you could always field a question, and I'm sure one of us or somebody in the National Ataxia Foundation will get back to you and let them know and let you know whether you would be eligible for a trial which is just, for example, opened up, for example.
All right. I have a question for Dr. Mcloughlin. It's from an individual who says, I am a participant of the CRC-SCA study, but due to COVID, some of my appointments were canceled or delayed. Is that information that I provided still important? Even if I missed some of my appointments?
100%. No, absolutely. So what Dr. Opal spoke about earlier is this idea of a snapshot in time, which is what you indicated. You've gone to one appointment for your CRC-SCA. A longitudinal study would be wonderful, but a single time point we can use based on your age, on your sex, on your background, on your genetic disposition. We can associate that with the larger pool from CRC-SCA, from ReadySCA subjects, from the U01 that I have for our University of Michigan. And I will say we had the same problem at the University of Michigan with our subjects. We went through a challenging time. Everyone did. Science was not while science was important through the last 2 years, it was absolutely shut down and challenged when it comes to clinical opportunities for us to make sure ataxia was moving forward. So I will say we are continuing to reach out to those participants from CRC-SCA, from ReadySCA, from the U01, and asking for them to come back in. And there's a safe space, at least at the University of Michigan. Right now, we have very few patients in the clinic for COVID-related indications, and we are enrolling. I actually had blood samples last week from 2 extra patients. So I encourage you to reach back out to whatever site you went through for CRC-SCA and say you're still very engaged and would love to continue participating, because every time point really does help us understand each of these diseases a little bit more.
Yep, and I like that you encourage patients to reach out directly to their investigators. These hospitals are very busy and these coordinators are very busy, and they would certainly appreciate patients reaching out directly and making it easier for them to get in contact with you and to schedule your follow-up appointments. So that's wonderful advice. And I'll send a follow-up to Dr. Opal from another person who participated in a CRC-SCA study. They said, I participated in the studies you mentioned, but I decided to opt out of the lumbar puncture. Will my data be considered incomplete? Is it still important?
Yes, first I want to say that everything which you do is important with regard to clinical trials, even the questionnaires, the functional rating scales, and the rating scales. So, and everything is done with your consent. There's no coercion with anything. So you're right. There's something which are part of the clinical trial, the rating scales, which are, if you choose to join the trial, that's part of it. Now, some things are optional, and you're right that the spinal fluid is optional. So, so you definitely can opt out of it, but everything else will be part of the clinical trial. Every piece of data will be compared to other parts of data. What we just have missing would be the spinal fluid. So your data cannot be compared to spinal fluid analysis or other aspects. Now, sometimes what we do, and not sometimes, but one of our goals is to do a cross-reference with different things. So in a CRC-SCA, there's a clinical rating scale, there's the imaging with MRI-based imaging, which is done at a few centers. And then there's this, the blood and the biomarkers, which will eventually be tagged on, because I have to say that some of these studies were only added recently. In the early version of the CRC-SCA, there was actually no spinal fluid, for example. So what we want to do is basically get a cross-reference across all these platforms. So we want to get a big picture of what's going on in the disease, with each aspect speaking to the other, so sort of fleshing it out. So obviously, the more information we have, the better. But as I said, if you don't have the spinal fluid, don't worry about it. We will take whatever we get, okay? Put it that way. And it's good for the field, it's good for patients, it's good for science to move it forward. So please participate, even if you don't want to do a spinal tap.
And we're almost at the end of the hour. Just a couple more questions. Can you participate in both the CRC-SCA and ReadySCA trial? And could you participate in a natural history study and a interventional trial, an actual clinical trial at the same time? I'll direct that to Dr.
We'll
McLaughlin.
alternate back and forth, absolutely. You can participate in CRC-SCA while participating in something like the UO1 that I have at the University of Michigan. So we do dual enroll in that sense. We don't take more sample. We just parse out the sample, what we require to send to this repository for BioSend goes for the CRC-SCA. But every one of these sites, there's always just a little bit of excess. And for my studies in our lab, we work with 10 microliters. So in that sense, that's a drop of a drop from a little, how do I say that? In my sense, a pipette, but a very small amount of fluid, a teardrop, we'll say, for us to be able to analyze information. And so having them already enrolled in our study means that I don't have to reach out to CRC, to the BioSend repository to ask for additional samples. And then those samples can be used for other programs across the country that are also looking into research. Now, interventional trials, as well as natural history, it gets a little bit more challenging based on that inclusion and exclusion criteria that I spoke about. Some natural history studies that are ongoing are a little bit more open. Others are a little bit more constrained in the sense of their exclusion criteria. So if you're actively taking a drug, then you might not be eligible. So again, reaching out to those clinical coordinators, it's a short phone call for you to define whether you would be eligible or not and how broadly you can spread the wealth, I guess, if you want to put it that way.
All right, well, thank you. We are at the end of our hour. So I just want to finish off by saying that if you are interested in participating in the CRC SEA or Ready-SCA trial or any other natural history study, I encourage you to get a little more experience with the clinicaltrials.gov website and as well as join the CORDS registry, because that's a good way to be contacted from researchers who are actively seeking patients and diseases beyond those mentioned in the CRC SEA site. You can also find more information on the National Ataxia Foundation website about active natural history studies. So again, I want to thank Dr. Mcloughlin and Dr. Opal for your time, and we really enjoyed your presentation today.